cell biology with numbers, and today I will discuss other properties of biological systems. So again, I continue with cell biology with numbers. Today is the third lecture, which I will dedicate to energy and time. Which are very important concepts in physics, but also in biology. Energy and time. This you can find in chapter 3, the reference I gave you. Chapter 3. You can find many interesting numbers related to energy and time scales in biophysics. The ones I'm giving you are the ones that I found the most interesting of all of them. And to motivate the problem that I will first discuss, I will give you a sketch about what happens in a cell. You have transport inside the cell. So we discussed in the previous lecture that there are some hollow tubes called the microtubules that span throughout the cell. These are like highways for some machines that move vesicles and objects and cargo. These machines are called molecular motors, which you can draw here in white. So for example, kinesin has like 2 legs. This trying to draw drawing. I'm not very good in drawing. This is called Kinesin Motor. Kinesin. It is a motor protein, which you can see as a machine that walks in a track. This is the track, the microtubule, and this machine is walking in a given direction along the microtubule. These machines can work alone or in company with other machines. So another 2 legs. This, for example, bent like this. This. And they have a very important function, which is cellular transport. So they can move objects that are much bigger than them, which is called vesicle. These are objects that have a membrane. So here is the vesicle. And these machines, which move in a given direction, are able to transport objects that are much bigger than them. So you can think about this like an ant bringing a piece of food in the cell, and this is done by this called Kinesin Motors. So, of course, these are very small. They do steps, and these steps, so this is like walking, stepwise, and these steps are quite small. So these steps are of the order of 8 nanometers for kinesin. But for different motors, typically, the step size is of the order of nanometers. And they exert forces. So this is dragging a vesicle, and this is pulling with a force. So if I give you a very simplified version of this process, it would be something like this. Like we have a linear track. This will be a very, very simplified version of the problem. There will be a motor which has 2 legs. This is the head of the motor. These are the legs. The motor moves in a given direction and can transport a vesicle. Usually it's much bigger than the motor itself. The motor is a very strong machine, and this vesicle exerts a force to the motor. Okay? But the motor, okay, you say, okay, I have a weight with me, and the weight is pulling me. In order such that the motor can move, it needs fuel. So you have to think about it like an engine. You put fuel in a car, and the car moves. So this doesn't happen by chance. It's not just random motion, but in reality, this has a chemical reaction here. This is powered by ATP consumption. ATP that goes to ADP plus P. This gives energy to the motor. So this exerts a net force such that the motor is moving in one direction. Okay. I don't want to enter too much in details because there will be lectures about this where I will explain this with very precise models. But what I want to give you a rough idea is about what is this size. So the motor is moving step by step. So in the next step, it will put this leg. This leg will come here. So its center of mass will move the period of the filament. And the period of the filament has a length that is typically of the order of nanometers. Okay. 1 nanometer is a very small quantity, and I said in my previous class, this is 10 to the minus 9 meters. Okay? It's a machine that is stepping. We do steps, we move 1 meter. These motors move 1 nanometer per step, and they exert forces of the order of piconewtons. Which is 10 to the minus 12 newtons, right? It's a very tiny force what the motors are doing. Now the question is whether the muscle of this motor has fluctuations or not. So if you take, for example, you take a car, you switch on the engine, you press the button, you can go at a very steady rhythm. You are in a linear track. That is, you just press the throttle and you go. So your motion, the motion of a car, you plot it in a linear track, x of t versus t. A car will do like this. Okay, assuming at the time 0 you already have the throttle in. So this will be the car. However, if you look at these motors, the motion doesn't look like this very much. How do you think it will look? Any idea? It will look like this? X of t? No. But any idea?
A step function?
Yeah, of course, there will be a step function. Well, this is a good thing. But this will also jiggle around, no? It won't jump like this. It will jiggle. So of course there will be jumps that are discrete. This is very important. But also you will have noise. Because this is not in vacuum. This is in a thermal bath. There are molecules constantly hitting this motor. This will jiggle. And depending how the jump happens, because the jump can happen slowly, the jump can happen like at once, you will have different possibilities. It could do like this. It could also walk backwards. Go back. This. This is the motor protein, the molecular motor. So the important thing is there is noise. Here, there is noise. Okay? Hello?
Sorry, I was checking the spelling.
Okay, fine. You can do it after the class. Try to keep focus, okay?
Yeah.
So the main point here is that this motor, it is affected by fluctuations. It's a very important word that you will see in my course. There are fluctuations. So this motor is in a thermal bath. There is water around the motor. There are water molecules here that are constantly kicking this motor, which are originating this is called the fluctuations. Fluctuations in the motion of the motor. So the question that I think is very pertinent to do at this moment, it is what is the energy of a thermal fluctuation. So how much energy do these water molecules have in the environment? The question is, what is the energy? Let's say this is wording the energy of a thermal fluctuation. Thermal fluctuation. So in other words, what is the typical energy of a water molecule near water? This is important because I am moving now on air, and air has fluctuations. So there are air molecules here. But these air molecules have an energy. So they are opposing my motion, but is that energy big enough to make me walk backwards? If I walk backwards, it's because I'm using my muscles. But it's not that there is a fluctuation that moves me backwards. Unless I'm in a very windy day in Trieste, you know, in that case it happens. But it is because you receive an air stream with high velocity. So now I'm talking about this in a quiet environment full of water. So what is the energy of one of these molecules? What is the typical energy of one of these molecules? Number. This is a bath in equilibrium. Bath in equilibrium. I think you had lectures of statistical physics. So you have let's say you have a cube and you have a lot of molecules. In a thermal bath at temperature T. What is the typical energy of one of these molecules? Epsilon?
Yeah.
Well, yeah, I call it epsilon. Okay, this is epsilon. But what is the typical energy? You have a cube, you have this, the water inside here. There are molecules in this bottle. What is the typical energy of each molecule in this? You should know. With 1 joule, they have 1 joule of energy. 1 joule. 1 joule is 1 Newton times 1 meter.
So it can't be that.
It can be that. What do you know? You know there's the temperature. So it should be related to temperature, no? No guess? Okay. The typical energy, the average energy, is of the order of kBT. Boltzmann constant times the temperature. That's very important.
That's not a number.
Sorry?
It's not a number.
The typical energy is kBT. kBT. You have a bath of water, and each water molecule, on average, has an energy of the order of kBT. There is a key-partition theorem. Key-partition theorem tells you, per degree of freedom, you have kBT divided by 2. This is very important. Okay. The typical energy of each of these molecules, typical means there is a distribution, there is a probability for each of these molecules to have a given energy, but the average one is around kBT. So, what is the energy of each of these guys? Typically, this has energy kBT. You call it epsilon, but you can call it A. It's of the order of kBT. And now my question is, now a molecule with energy kBT comes to me, and I'm moving. Is this energy higher than my energy when I move? My energy when I move is mass velocity squared, kinetic. See? In principle, this should be very small, no? Otherwise, an air molecule will kick me back, which doesn't happen. Now let's see here. Let's see here. Okay. So let me calculate what is kBT. So the main point is kBT. How much is kBT? So this, how much is kBT? This is my question. Okay, kBT. So, okay, if I ask you this, you should know Boltzmann constant is a constant. It doesn't depend on the temperature.
Yes.
But if I ask you this, I should tell you which is T. So T, I will assume this is in around 300 Kelvin. This is typical value for it's like 35 degrees Celsius degrees. Okay. So for this temperature, we have Boltzmann constant times temperature. How much is the Boltzmann constant? I tell you, 1.38 times 10 to the minus 23 joules per Kelvin. This you can find in any physics book. It's a well-known number. And the temperature is 300 kelvins. So you kill the kelvins, and now you have 10 to the minus 23 times 100. It's 10 to the 2. This gives you 10 to the minus 21 joules. Okay, so you could say, fine. This is around 4 times 10 to the minus 21 joules. Fine. But what are the joules? The joules are, this is 4 times 10 to the minus 21 newtons meters. 1 joule is 1 newton times 1 meter. But this motor has the force of piconewtons and distances of nanometers. So I would like to compare with these units. Then what I will do is say 1 newton is 10 to the 12 piconewtons using this. And 1 meter is 10 to the 9 nanometers. So I keep the newtons, keep the meters, and I have piconewton nanometer. And look at this, because 10 to the minus 21, 10 to the 12, 10 to the 9, this is 10 to the 21. So this becomes 4 piconewtons nanometer. This is a very important number for you. Very important, because look at the KBT, one water molecule has this amount of joules, it's very little, but in piconewton nanometer, it's very easy to remember this number. 4 piconewton nanometer. Okay, and now I ask you, is this important for this motor? This or this not?
Excuse me, this is the net energy that is exerting in one direction?
In any, because water molecules are coming from the left, from the right. So we have a bath, like here in a cube. So you have the motor, and there are, you have to think there are millions and millions, much more than millions. There will be a Avogadro number of water molecules.
It is somehow having the assumption that they are exerting all of internal energy as a force.
Yeah, because they have kinetic energy. Water molecules have velocity. So the energy will be roughly their kinetic energy. So they have a kinetic energy of this value. So it means that it can come one water molecule with a given velocity and hit the motor. And the motor moves back. But they can come 2 at once, they can come 3. It's very random. This. What I want to know is, on average, when a water molecule hits the motor, how much energy is given to the motor. And this of this order. 4 piconewtons nanometer. And this is important for the motor, because the motor does forces of the order of piconewton and moves a nanometer. So if the force is, for example, 0.1 piconewton, and this is 1 nanometer, you will have 0.1, and the thermal fluctuation will be 4. So it will be 40 times stronger. So this tells you that thermal fluctuations are important for the motion of a small biological system. So just to give you an idea, it implies that the work done by a motor divided by the energy of a thermal fluctuation, which is KBT, will be of the order of 1 piconewton nanometer divided by 4 piconewton nanometer, which is 0.25. So if the force is 1 piconewton, the work done by the motor in one step is 1/4 the energy of the thermal fluctuation. So this will be a motor that is moving backwards a lot of times. Okay? It can move backwards a lot of times. But if you have another case, okay, this is another example, example 2, in which the force, for example, is, we could say the force is, 20 kilonewtons, for example, and the length is 1 nanometer, then this example is different because the work of the motor in a step divided by KBT will be 20 piconewtons nanometers divided by 4 piconewtons nanometers. So this will be 5. Here, the work done by the motor is 5 times larger than a thermal fluctuation. So this will correspond to an example like this, that there are back steps, like here, and this one, which I show it in green, will be a case where you will do like this. There will not be many back steps, right? This is to give you an idea. So the main point here is to know that KBT is not that small for biological systems. Okay, it's not that small. Of course, if you, instead of having one motor, you have 100 motors pulling together, then the fluctuations will be less important. So this will scale with the number of motors, right? And with the forces and with the length. So this is to give you an idea of the order of magnitude. Any question? It's a very important number to know. Right? Yes. Of course, this is for T 300 Kelvin. If I put the temperature of the sun, so you will put here thousands, it will be different. Okay. But I'm doing the typical temperature in water, 300 Kelvin. All right. So if this is clear, it is good. I'll show you another interesting number related to KBT, which is concerning also transport in the cell, but at a larger scale. So the next example is for cell motility driven by polymerization of actin, which I discussed in the previous lecture as well a little bit. So, the question that we try to solve now is the following: is how much ATP is required for cell motility. For cell motility. Right? Of course, I cannot make this question like this and say how much ATP is required for cell motility. There are different types of cells. Each cell has a different size, different forces, different mechanisms to move. So I have to give you a model if I ask you this. And the model and the data that I give you is the following. So we have a cell of a very irregular shape that has a height of 20 microns, okay, and that it has a cytoskeleton. So remember, this is called the cytoskeleton. That is made of actin filaments. Actin filaments. And this is moving. So I will do an approximation here. I'd say, okay, of course the actin, the cytoskeleton is like a mesh of it's a network of filaments. They are all moving. I mean, it's not so regular as what I will do now. But in the approximation I will do is that I will assume that there is one side of the cell growing in a way that the filaments are all pointing in the same direction. This is a very crude approximation, okay? And that there is a homogeneous density of filaments. This is another assumption. And each filament, all of them, are growing at a given speed, which is, the speed is 0.2 microns per second each filament. Okay. Per filament. Okay, filament has no units. I could put nothing here. Fine. So each filament is growing at this speed. So you have like a rope, one-dimensional rope that is moving, growing at 0.2 microns per second. This is one of the data. This is the other one. The size of the cell, the size that the side length that is growing as 20 microns of length, and then there is a density of filaments. The density of filaments is 200 filaments per micron. Okay. This tells you there are 20 microns. In each micron, there are 200 filaments. This is the density of filaments. And with this, okay, I will need a couple of things more, which I will give you later. One is what I can give you now. So how is actin polymerized? So I'm putting a very simple sketch here of a polymer that is growing, but this has a molecular structure. So if you zoom in, here there are monomers. Okay, I'm zooming in here. There are monomers. So this would be the actin filament, the leading edge at one time, but when you incorporate a new monomer, it will look like this. Okay? And then there will be a new monomer. Okay? This is the polymerization of actin. This is, for example, time equals 0, and this is time equals delta t. At delta t, we have incorporated a new monomer. Right? And this is done thanks to a chemical reaction. So when you incorporate a new monomer, you're also using energy. The energy comes from ATP. So there is a chemical reaction for this to happen. This ATP, adenosine triphosphate, going to ADP plus P. So you take this molecule and you break it into ADP and P, and this gives a lot of energy. Free energy, actually. So the free energy for this reaction is around minus 14 kVts per monomer. Okay, to incorporate one monomer, you use 14 kVts of energy. So now the question is, why I put here a minus? Is this by chance, or is there a reason?
Because ATP is being broken down.
So let's think about it, about this problem. So
And energy is like
the ATP is the energy, and when you have this transformation, the ATP, you have a loss of energy from ATP.
there's a loss of energy. This is the main point. It's an exothermic chemical reaction. So if you can draw the free energy, G, versus the chemical reaction, you will have something like this. You can think as a physicist as this as a diffusion process. So like you have a particle in a potential, which is the free energy landscape. This is the reaction coordinate, and then you go from ATP to ADP plus P. The story is that you go from a state of high energy, high free energy, to a state of lower free energy. Okay? That's why this is negative. because in reality this is the free energy of ADP. That's P minus the free energy of ATP. Okay, the free energy at the end is less than at the beginning. That's why you have a negative number. You're going to a state of lower free energy. These are the chemical reactions that often happen. If I had the landscape the other way, delta G will be positive, and this will be a reaction that doesn't happen spontaneously. Okay, it's an exothermic reaction. Chemical reaction. And this is the Gibbs free energy state. Energy change of ATP hydrolysis in actin polymerization. Actin polymerization. Okay, in other reactions, ATP is used in a different way. Here is 14 kV different. So in principle, this is almost all you need to solve this problem. We need also to know what is the length of a monomer. So what is the length of a monomer? And what is known is that two monomers are 5 nanometers. So one monomer is 2.5 nanometers. So with this 1, 2, 3, 4, 5 numbers, we will calculate how much ATP is required for this cell to move. So what I want to solve is how much ATP I need per unit of time. Okay? So in 1 second, how much power I use, how much energy do I need, what does the cell need to produce, to move at this speed in the living age. So let me go step by step. The first thing that I will calculate is how much actin is produced per unit of time. This I call the actin polymerization. You will see that this problem is not so difficult once you know all these numbers. Okay. Actin polymerization. Right. So to do this, we know first that we are moving 0.2 microns per filament per second. Next, okay, these are how many microns we move per second in a filament. Now we should know how much actin is this. So we know each monomer of actin is 2.5. So, but this is in nanometers, and that we have in microns. So we have to adapt this. So we will say first that 1 micron is 10 to the 3 nanometers. Now we know how many nanometers per second is advancing this filament. And I want to know now how many monomers we are adding per second. Okay? For this, I will use this number. So each monomer is 2.5 nanometers. 2.5 nanometers is 1 monomer. So, very easy, 0.2 times 10 to the 3, 200 divided by 2.5. You should know. Well, actually, why do we have 200 divided by 2.5? Well, it's around, imagine you cannot calculate this, it's around 100 monomers per filament per second. Okay. 2.5 to, I'm doing an estimate. So maybe it's a bit less, maybe 75, maybe. But remember, we are talking now about orders of magnitude. The number I will give you at the end will be 10 to the power something. So it doesn't matter. I'm doing this. Similar equal. This means if you don't have a calculator, you can do it. Right? Okay, so now we say there are per second 100 monomers included here in each filament. But now I'm asking the ATP in the entire cell. So I have to multiply by the length. This would be first thing. So we will multiply by number of filaments in the cell, and then we will know how many monomers per second in the entire cell. And then what do we need? What else do we need? I'm asking about the energy.
You need the force.
Not the force. I'm telling you here
Or
the free energy. So this is how much energy do you spend in adding one monomer. But now I will say this is like building a wall. So you need to put a lot of them, not only in one direction, in all of them. That's what I'm calculating now. First I'm telling you how many I add here, and now I multiply by the size of the cell. So let me show you how this can be done. So I will now calculate the ATP consumption rate, which is what I want to calculate from the beginning. Consumption rate. So this will be the following. Let me call it W dot. Why W dot? This is like work per unit of time. How much work I'm doing? Work is energy in this context per unit of time. This would be, okay, first of all, I have 20 microns of them. So 20 microns. I multiply this by the density. So we have in 1 micron, we have 200 filaments. So what do I have until now?
number of filaments.
There are number of filaments. So I'm counting this number of filaments. I multiply length times density.
It's a 20 micrometer is the length of each filaments.
No, be careful. 20 microns is the length of the entire cell. The length of the entire cell. And this is the density. So length times linear density gives you a number. Okay? Up to here, this is the number of filaments, all this, the number of filaments that are growing in the cell. Then I need to know, okay, I have this, which tells me we have 100 monomers that are incorporated per filament per second. This is the actin polymerization rate. This one. So now I kill this, and I have monomers per second. So in the entire cell, how many monomers I am adding per second? It's good. And then, well, it's almost ready, because now I should say each monomer,
Needs that much energy.
per monomer, it needs how much energy? So around, again, this is an estimate, around 10 ATPs.
How do we come with the energy?
From here, per monomer, this is the chemical reaction, ATP plus P. This is the chemical energy that I use to incorporate one monomer. Gives you energy change of hydrolysis in actin polymerization.
Yeah.
Okay? Each step in actin requires 1 ATP. There are other motors that use less energy, and there are other motors that use more
There
energy.
are 10 steps.
In each step, per monomer, adding one monomer requires 1 ATP. But there could be a motor that is more complex and requires more ATP.
No, but how do we get 10 ATP?
Because I want to do an estimation. So
So We're just assuming that.
Yeah,
Okay.
because you see, I have 10, 100, 110. So I will get a 10 to the power something.
Okay.
So it won't matter if I put a 4 here.
Okay.
It won't matter. This is because I'm doing an estimate, okay? Just this. So now, the nice thing is we can do this calculation. First of all, we have ATP per second, and we have 20, 200, 110. This you should be able to do it without calculating. Okay, 20 times 200 is 4, 10 to the 3, 10 to the 5, 10 to the 6. So it's 4 times 10 to the 6 ATPs per second. That's the answer. So this cell needs 4 million ATPs per second to move at this speed. It's a lot. It's a lot, but now, okay, actually in my notes I have a mistake here because per monomer, yes, there's a mistake in my note, but so now we are doing it correct. So this is 4 million ATPs per second, and now I will ask how much is this in, okay, so we have W dot is 4 million. Actually, ATPs per second. No, sorry. This is incorrect. Incorrect. In each step, be careful, in each step I'm using 1 ATP.
That's
Not 10, not 10, okay. Each step is 1 ATP. So 10 to the 5. 10 to the 5. Why this happens to me? Because I work in thermodynamics and I'm always using KBT. So I always think in KBT. But per monomer, I use 1 ATP. So this is correct. And now I'd like to know how much is this in KBTs per second. So to do this, it's very simple. So I have 4 times 10 to the 5 ATPs per second, and I have this. So I have that 1 ATP requires 14 kVts of energy. So this is quite simple and gives you around 5 times 10 to the 6 kVts per second. This is another valid option. So the first option is I tell you how much ATPs I use per second, and this is how much energy in comparison with thermal. You see that the motor, to make a step, takes 1 kVt, more or less. A cell, to move, requires millions of kVts. It requires much more energy than one single motor to move. This is important for you to know. This is a bigger object, it requires more molecules, it requires also more energy. So, as expected. Okay. This is, okay, nice. And now, if you want to compare with our scales, macromolecular, macroscopic scales, you can realize that, okay, how much would this be in terms of, okay, you have a car and you have the power of a car, W dot. What is the unit you use to measure power or in an electric device or in a car?
what?
Watts, no? Watts is joules per second. So it would be nice if I can say how many watts I will need to make this work, no? How much is this in watts? So this is in the scales we care about, kVts, ATPs per second. We care about these scales, but if we want to do this in macroscopic scales, let's say human scale, okay? The unit of power will be watts, okay? So we can try to convert this to watts, and we see that this will be, let's see, how much is 1 kVt per second? 1 kVt per second is, remember, 4 times 10 to the minus 21 joules per second. this is what I did in the beginning of the lecture. 1 kVt is 4 times 10 to the minus 21 joules. And this, you can write it as 4 times 10 to the minus 3 AW attowatts. So this is called, okay, 1 attowatt. Okay, this is attowatt is 10 to the minus 18 watts. So 1 kVt per second is 4 times 10 to the minus 3 attowatts. It's very small. So W dot, if I don't remember well, okay, we have 10 to the 6. 10 to the 6 times 10 to the 3, the minus 3 is 10 to the 3. 5 times 4 is 20. This is 2 times 10 to the 4 attowatts. Okay, I give it as a curiosity. This is not so important. It is just to show you that this is small compared to what an electrical device needs, a computer and so on. This is small. And I think with this we can do a break. You're all fine, unless you have questions. Okay, maybe the break. All right. So go with the next topic about energies times. Well, I'm talking about energies. In particular, I'll discuss first with an example. Then I will pose the question. So the example will be about transport through membranes of ions. So here is a nice drawing. We'll try to do some nice drawing of a channel. This is a channel in a membrane. Okay, the drawing is not so nice. But the channel is a structure like this. This is an open configuration of a channel in which ions, this is the membrane, plasma membrane. This is the channel. Okay, this could be, for example, the extracellular medium. Extracellular medium. And here the cytoplasm, the inner part of the cell. I call this an ion channel. Which is, you can think about it like a gate that opens and closes during time. And this gate lets ions, which are electrical charges, pass through it. For example, you can have a positive ion coming like this. And the only mechanism of this channel is opening and closing due to thermal fluctuations. So the channel can be open and closed, and this happens randomly. I will give a very detailed lecture about these channels, but the main thing you should remember is what happens when you have, okay, this we will say this is called so-called passive transport of ions, in which there are some physical laws that rule the current through a membrane when there is a concentration gradient. So for example, imagine you are in a medium which has a lot of positive charges, and the cytoplasm has few positive charges. What will it happen when you have this gate open? Exactly. Boris is doing like this.
Yes, ions will go in.
They will go in. So in the end, there is a exactly. That's it. There is a physical law. It's called Fick's law, which tells you that there is a current of ions that is proportional, so minus fusion coefficient times, well, we put gradient of C. So it goes against the gradient. So you don't go up here, you go down here. There is a lot of concentration here, so there is a current moving you down here. Okay, this is the slope of C of x. This is against the slope. So as Boris is doing, like this.
But why is there a negative sign?
Because, okay, so if you have something like this is C of x versus x. This is in 1D channel, and you have like this. What is the derivative of C? The derivative is positive. So if you do C prime x, okay, in 1D this will be minus D C prime x. Okay? C prime here is positive. So C prime, this is 0, this is x, C prime is positive. But the flux is not going in this direction, the flux is going in this direction. So it goes against, okay, it goes in this direction. This is why there is a minus. Nothing else on this. This is called Fick's law. So for this, we know very well what happens when you open a gate and ions pass because of the concentration gradient. However, there is a different situation. Okay, let me erase this because I need some space. There is a different situation to this one, which is actually what happens many times in biology. It is the so-called the ion pump. So you can have in biology machines that move ions against the gradient. Okay, this is another story. It is okay. We'll do a similar sketch, and I think this will be helpful for you if I do. Okay, I can send you later from where I took it. There is one part like this. There is another part like this. Sorry. Okay. Then there's the membrane. And now let me say that there is an element that is more complicated. That's this type of shape. Okay, let me try to imitate this. Okay, something like this, which can rotate. For example, in this direction. With the help of some energy, okay? This could be rotating due to ATP consumption. So it's an element that does like this, okay? So an ion can come here, then it rotates and passes. Okay, and this could be, for instance, selective to the passage of different ions. Okay, this is the membrane. This could be a very selective example. Imagine this is sodium ion, sodium passing like this. And this could be, for example, the sodium-potassium pump that lets the sodium pass like this, and the potassium, this is K, come like this. This, for example, is a cavity that has the size of potassium. This is a cavity that has the size of sodium. In this way, you would control the passage of ions and let this work. For instance, against a concentration gradient. You could have a lot of sodium here and still pass the ions of sodium upwards. But for this, you will need a mechanism, something. You need an energy source that moves this.
I think it's 3 and
Well, the main point is you need a source, so you need something which could be, for example, ATP consumption. ATP going to ADP plus T. So this type of motion, okay, this the one here, this is called an ion channel. So I put it in this color. And this is called an ion pump. This is just a sketch. And this generates passive transport, the ion channel, and this one we say it generates active transport. And of course, the physics running active transport is more difficult than fixed law. This is just for you to see that in biology there are more complicated mechanisms of transport than just pure diffusion. Right? This is just a motivation, but yes.
Are we inside the microphone here?
Sorry?
Are we inside the microphone here?
Well, mitochondria have also membranes and they have ion channels here.
These are neurons.
What?
Specifically neurons.
This can be in a neuron, for example.
Yeah.
But
it can be other things.
okay, I'm not going to discuss very in detail much of the biology. For you, just the main concept you need to keep is that there is transport in biology that can go against the concentration gradient because of the usage of chemical energy. This is the main point. This is called active transport. And in general, there is a big field called active matter and active processes in physics, which is taken out of researchers. There's a lot of people doing research on what is called active matter and active process. It's a very active field, actually. Okay. So, before going to this, I'll first discuss one model of an ion channel. I will focus on these ones, but you just have to keep in mind that there are more complicated ways of transport in biology. So the question I will discuss now is the following: is what is related to this? Is what's the electric potential difference? Electric potential difference across membranes. So this is the question and focus part of the rest of the class. And I told you more or less how big is a membrane, how thick is a membrane. So we have a membrane with phospholipids, and in the previous lecture I told you about the thickness of this. You remember? 4 nanometers, exactly. This is around 4 nanometers. However, it is important to know that this is not just soft matter. There is also charges in the membrane. The membrane is charged. And what we will do is to use a very simple picture of the membrane as a capacitor, which you may remember from your physics courses. A capacitor was something like this, which has a positive charge, an excess of positive charge in one side, and an excess of negative charge in the other side. You remember this from your physics lectures. So now I'm going to think about a capacitor which has the size of a biological membrane. So the main idea is that it will be a capacitor of this thickness, the thickness of a membrane, and that it will have a given voltage. Remember, the capacitor has a certain charges accumulated in its sides, and this is connected to a voltage difference. So here there is a charge, here there is a voltage, which you can call it in many ways. You can call it delta V, and this has a capacity, C. Okay? You remember this from your... Alright, so having said this, what it's important is to know what is the the membrane potential, the so-called membrane potential. So the membrane potential has been measured in many experiments. Potential, and it's thought, let's say that I give you this quantity, I call it U, and I tell you this is around 100 millielectron volts. What does this mean? Any guess? Millielectron volts.
You want to compute that?
I'd like to know, I ask you, what does this mean? Millielectron volt.
It's the differential of potential between the condensator.
So how much is this in volts?
1 electron volt equals to 1.6 times.
No,
30.
no, no. Because milli is 10 to the minus 3.
Yeah.
This is 10 to the minus 3 volts. But there is this E. So electron volt is a unit of energy, not of a voltage. So I will give you a reminder about what is the electron volt and how much is an electron volt. So let me ask how much is this? Okay. How much? and what? What is this exactly, no? So what is the electron volt? Should remember mainly that the electron volt is how much is the formula. You have a charge, let's say you put a potential, delta V, that is 1 volt here, plus minus, put 1 volt. And you put, okay, this will be minus, this will be plus. So the electron volt is how much energy gains an electron when it moves, when it's subject to a voltage of 1 volt. Okay? So the electron comes here and feels all these negative charges and is traveling through this part. So the electron volt, 1 electron volt, in reality is a unit of energy, not of a voltage. Okay, so it's 1 coulomb, which is the charge, the energy, times 1 volt. Okay, and this, all in all, it is very important that you know how much is this in. Okay, this would be sorry. So the energy, it's, okay, this is the charge, Q, and this is delta V, okay? Q times delta V is energy. And now, how much, okay, sorry, I did a big mistake, because I put here 1 coulomb. What is the charge of an electron?
1.6.
Very good. You have to be awake in the class, otherwise these things happen. So, 1 electron volt is the charge of an electron times delta V, which is in this case 1 volt. This is the energy change in an electron when it's subject to this voltage. The charge of an electron is 1.6 times 10 to the minus 19 coulomb. Okay? And the corresponding delta V is 1 volt. This is the definition of the electron volt. Okay? And then coulomb times volt is joule, exactly. So this is 1 electron volt is 1.6 times 10 to the minus 19 joules. Right? And this we can now transform in cavities, actually. So the first thing is this one. Now we know what is 1 electron volt. Okay, this you can find it everywhere. And now I tell you, the membrane potential is 100 millielectron volts. So this is 100 millielectron volts, and then we say 1 electron volt is 1000 millielectron volts. This is what you were not saying correct.
Yeah.
Milli is 10 to the minus 3. Okay? 1 milli electron volt is 10 to the minus 3 electron volts. So, like this. So up to now we have 10 electron volts. 0.1 electron volts. Sorry, I'm making a big mess now. 0.1 electron volts. And now we can say that 1 electron volt, the unit of energy, is 1.6 times 10 to the minus 19 joules. Okay? Now we have the energy in joules. And then what we can do, let me see if I have it here. What we can do is to write this in units of kVt. So this would be, remember kVt is 4 times 10 to the minus 21 joules. This goes with this. This means that I will write U in kVts now, right? So what I get is, today I'm very lucky, everything is equal to 4, I don't know why. So this is 4 kVts. So in a membrane, the potential that you put is around 4 kVts. The electrostatic energy across a membrane is around 4 kVts. This is a rule of thumb, okay? It's 4 kVts. This means that when they give you an energy in milli electron volts, you can say that 1 kVt is 25 milli electron volts. Okay? This is a conversion factor you can use from here. But in a membrane, we say 4 kVts of electrostatic energy if we think in the membrane as a plane capacitor. Right? So, alright. And then, okay, this is around 4 kVts. And to give you an idea of how big is this, so the electrostatic energy for DNA rupture to break DNA is much higher. So this is of the order of 400 volts. Okay, so the energy that is here in the membrane is much less than the energy needed to break the DNA. So if you put, sorry.
I didn't understand that 1 electron volt came to 4 kVt, and here 1 kVt is equal to 25 millielectron volt.
Be careful. I said that the energy here, here there are 100 millielectron volts. So if I would tell you this before, you would have the answer immediately. This is the answer. This is 100. Okay? 100 millielectron volts are 4 kVts. 1 millielectron volt are, okay, I did a mess, I think. 1 second. 1 second, because I think. Yes, 1 kVt is 25. Yes, and 4 kVt is 100 millielectron volts. Exactly. Yes. All right. So this is the electrostatic energy. But now I'm thinking about, let's say if a poor electron comes here, it will suffer an electric field, no? So one important question would be, how much is the electric field? Electric field here. So here there's an electric field. Let me put it in yellow. So if we have the positive charges here, and the negative charges here, where is the electric field pointing? In which direction? Left to right? Okay, who thinks it goes in this direction?
It actually goes in this way.
Boris is doing this way. And you? The electric field is this way or this way?
It should
This
be,
way.
by convention, it should be this way. Actually,
It is this way because the electric field is defined as the force done on a positive
yes,
charge
yes. By
of
convention.
1 coulomb. on a positive charge. It's the force
Yeah.
done on a proton. If you put a proton here, it will move in this direction.
Yeah.
Okay, but we are always thinking about electrons, so
Yes.
the electron will move in this direction.
Yeah.
So now the question is, what is the intensity of this field? Intensity of the field, electric field, it's just the intensity of the field is U divided by D. So the electrostatic energy in this capacitor divided by the thickness. This is a plain capacitor. It's very, very simple, big simplification. And then we say we have 100 millielectron volts divided by 4 nanometers. So we have 25 millielectron volts per nanometer. This is the magnitude of the electric field, and for curiosity, I will change this in other units. Okay, this will be the answer, but I will compare this with something bigger. So I will change this into, I will first change millielectron volts to kilovolts, kiloelectron volts. So 1 kiloelectron volt is how many millielectron volts? Kilo is 10 to the 3, milli is 10 to the minus 3. This is 10 to the 6 millielectron volts. And nanometers, I will put them into kilometers. 1 kilometer
10 to the power 3
is 10 to the power 3 meters.
meters.
So, how many nanometers? 10 to the 3 times 10 to the 9.
Yeah.
10 to the 12. 3 plus 9, 12. 10 to the 12. 12 nanometers. So there is here. Okay, actually, this is very strange. The calculation here was totally different. No, sorry. I wanted to pass it not to kilometers, but to millimeters. So forget about this, and you will see why. So I will do per millimeter. So 1 millimeter is 10 to the 6 nanometers. Okay, these two. Okay, this goes with this goes with this, and 10 to the 6 goes to 10 to the 6. So I have also 25 kiloelectron volts per millimeter. It means per millimeter of membrane, I have 25,000 volts. This is really a lot. A battery, if you go to the supermarket and buy one, I don't know what's the name, I know it in Spanish, one cell is volts. Here it's giving 25,000 cells per millimeter of membrane. This is large. Okay, and I will compare this number with now something a bit. Okay, this is a very bad drawing, which will be. This is a cloud. This is the ground, and in the middle there is a thunder. Okay. Okay, there's a thunder. So, I'd like to know what is the electric field produced that originates a thunder. So you can look around, and the main point is the following. What is the height of a thunder? More or less. A cloud, what is the height of a cloud? You have taken planes to come here. The clouds were below you at some point.
Yeah.
So, more or less.
We didn't measure.
You should know this, no? This feels bad not knowing this. When you take a flight, you can go above the clouds, no?
Yes.
The flight, what is the height of the flight? You should know it. Feet, meters? Around, you can go, this is most important, around 10,000.
Yes.
10,000 meters. And okay, so you see the clouds below you, so let's do a very rough estimate. 1 kilometer, okay? Well, 1 kilometer. 1,000 meters.
1 kilometer?
It could be more. Could be more. 1,000 meters.
It depends on the cloud.
It depends on the cloud, okay?
It does depend on the cloud.
Let's be conservative.
Yeah, very conservative.
let's be conservative. Okay, we can do 10 kilometers, okay, but it's not that much because you don't have a cloud when you are in the top.
Well, maybe if we're like on a hill.
No, on a hill you.
We're already like.
Let's try like this, then we see. Then, what is important is what is the voltage difference. So there is a voltage difference here. Okay, this is difficult to measure, but I found this somewhere, and it was 3 megavolts for a thunder. 3 megavolts. Mega is a lot, so it's 10 to the 6. So the electric field for a thunder is around 3 megawatts divided by 1 kilometer. And now let me compare. So, okay, this will be MeV. So this is the energy given to one electron falling in the cloud. So I will pass mega electron volt to kilo electron volt. So mega is 10 to the 6, kilo is 10 to the 3. So this is 10 to the 3, kilo electron volt. And I will pass kilometer to millimeter. So 1 kilometer is 10 to the 6 meters. So it's 10 to the 9. 10 to the 6 meters is 10 to the 9. Sorry, 1 kilometer is 10 to the 6 meters. 1 meter.
1 kilometer.
1 kilometer is 10 to the 3 meters.
3 meters.
1 meter is 10 to the 3 millimeters. So 1 kilometer is 10 to the 6 millimeters. You always have to think this very carefully. 10 to the 6. So now mega goes with mega, kilometer with kilometer, and I have kilo electron volts per millimeter, and I have 3 times 10 to the 3 divided by 10 to the 6, so it's 0.03 kilo electron volts per millimeter. And I put a big exclamation mark for you to realize it's much smaller.
Much smaller. And then what's happening inside the pot? Wow.
It's much smaller. So this is interesting to check.
Yes.
That the cell has a very strong electric field for compare even to a thunder. So good to know. So of course this in the end what counts is also the electrical current. How many charges are passing per unit of time. This is really the important thing. What can kill you at the end, no? The current, not the electric field. I don't know if we have more time, but there's something else I wanted to tell you. I don't know if I have time. It's simple actually. Okay, you got this, it's fine. I'll try to discuss the last element that I think it will take me around 15 minutes. So I hope I'm not taking out this room before I end. Be careful because the clock here is not going well. So if you look at this, you will always see.
All of the clocks in the building are the same.
Yes. So be careful with this. Okay, so I calculated here the electric field, and I will finish just saying how many protons are needed. To sustain this electric field. So what I will do is the following. So the question is as follows. So we are saying there is a voltage and there is a cell, there is a plain capacitor like this. Okay, now I'm doing a different drawing. So I'm thinking the cell has a surface. It's a plain capacitor. Okay, give me a second. And they'll say, okay, they'll say is a big simplification because the cell will be like this, and the cell has a surface. Surface is all around, and the surface area is of the order of 5 microns squared. Okay, which will be 5 10 to the minus 12 meters square, right? And I'm doing this approximation. So now I have this electric field in this plane capacitor. The approximation is a plane capacitor. In which we know the electric field, which is this value. We know the area, this is A of the capacitor, and another thing that we know in cells is the so-called relative permittivity, which is of the order of 2. I don't know if you remember this. Yeah. This is the relative permittivity with respect to vacuum. So the main formulas you need to solve this problem is the following. First, you need to know that the capacity is the charge divided by the electrostatic energy. So we know this, well, we will know this. We know this from there, 100 millielectron volts. And this Q will be the number of protons times the electric charge. This will be 1 coulomb, and this is U. Now, this will be easy because I will solve MP, and it will be Cu divided by E. But the point is what is C. So I didn't tell you what is the capacity. We are doing now an electrostatic problem, electricity problem. We have a plane capacitor of this area, of this relative permittivity, etc. So the question is, what is the capacitance of this type of capacitor? Do you remember from your physics lectures? Okay, you know the formula? C is, you can guess, it's fine. Don't be afraid of making a mistake. Okay, for a plane capacitor, the capacity is epsilon r, epsilon 0, area divided by length. That's the capacity of a plane capacitor. If I tell you this is a sphere, the formula will be different, okay? This is the capacity, capacity or capacitance? I'm sorry. Capacitance. Capacitance of a plane capacitor. Plane capacitor. It has this formula.
What's the difference between capacitance and capacity?
Yeah, it's what you find in the dictionary, no? I think I'm Spanish, so for me it's I don't even remember in Spanish. El condensador y la capacidad. For me it's the capacidad.
For me, it's studied in Persian, and this, I only heard one word, and I hear two different words, and I was.
If you want, you can come here and write it in person. Later. I don't know write in person, so for me it will be very difficult. Okay, so we have this formula, and now the main thing is we have to solve. So MP will be Cu divided by E. So this will be C is epsilon r, epsilon 0, A divided by D, U divided by E. Okay? Right, so you can put the D in the denominator. So this becomes epsilon r is 2, epsilon 0 is 8.8 times 10 to the minus 12 coulombs coulombs meter volt times, okay, I'm getting out of space, times the area is 5 times, here it's useful to have it in meters because epsilon 0 comes in meters, minus 12 meters squares times U, which is 0.1 volt, is 100 millielectron volts, so we can write it like this. And then it's EV. So E is the electric charge, 1.6 times 10 to the minus 19 coulombs, and D is 4 nanometers, 4 times 10 to the minus 9 meters. Okay, you see that I put everything in the international system because I'm taking this constant in the international system. So you see, coulombs goes with coulombs, meter squared, this meter goes here, so meter squared, the meters go, and that's it, volts go with volts. So number of protons shouldn't have units, as it is. And we get 5,500 protons. So the cell needs around 5,500 protons to produce this electric field. This is the answer to the question. And I finished just by saying that, for example, in E. coli, there are around 10 to the 8 charges per cell. So here we say to maintain the potential, the membrane potential, we need all these protons. So it means that MP, the number of protons, is around 10 to the minus 4 in total, the total number of charges. Just as a curiosity for you to see. So this gives us the solution. As you can see, we can do analogies between biological systems and capacitors, channels, many things in physics, and there are some curious and interesting comparisons between the electric field in a membrane and created by a thunder, and I hope this is interesting for you. Of course, if you go to chapter 3 in the book, you will find other interesting numbers. And well, if you have any question, this is the moment. If not.
Are we going to use all of these numbers to do something else?
Yeah, of course.
What are you going to do?
At some point, yes. I don't want to do a spoiler of the course. He will come slowly, and no, this is just for you to know scale. So it's more general knowledge about the biological systems and about scales and sizes, energies, and so on. Later on, this will be a motivation to introduce later the stochastic process, which will be a bit of math, and then we will do models with the stochastic process. So first you need a motivation, we need math of systems with noise, then you need this math. This is a very powerful tool for you, and then we will do something with it more related to research and models of biological systems. So it goes, the course goes smoothly, but the difficulty will increase soon, so don't, it will be getting difficult but also more interesting with time. So I hope it's good. Other questions?
What means 10 power minus 4?
Yeah, because this is 10 to the 3, well, almost 10 to the 4, almost. This is 10 to the 8. So in total, in the cell, there are 10 to the 8 charges. And we only need...
Where is the total?
This is taken from the literature. This has been
You
measured.
said the total charge of whole cell.
Yeah, the whole cell has 10 to the 8 charges.
Okay.
But you only need 10 to the 4 to create the memory potential. So you need to invest around 10 to the 4 of the total. A small fraction. I just did this divided by this. And roughly, of course, I'm putting this. If I want to be precise, I do the exact calculation.
And the number of protons here means the number of ions?
Well, the number of protons, because here I said each proton has a charge E. If I would say, okay, I want to build a membrane only with sodium, the sodium has a different charge. And then I put something else here.
Okay. And something I noticed that we draw that the active transport import the potassium and export the sodium. Why you do that? Both of them have the same charge. Both of them are positive charge.
Yeah, this is a good question. Actually, I don't remember the answer, but
It's because we have to, we need the potassium, I think, inside for the neurotransmitters for synapses, as far as I remember.
As far as I remember, it was something related to cell physiology. So the cell needs some ions to maintain its activity, and the other ions are used for energy supply. So it's sometimes, for example, you need transport of calcium ions in the transduction of the sound. This is very important. So you need that calcium enters in the cell at some point, and this is done also with active process. So depending on the ion, each ion has different biological function, and sometimes you need this ion to be transported against the gradient of concentration. So you need sometimes, the cell is in a hurry and needs to produce a lot of ATP, for example. It needs to also to send signals and say, okay, I'm here and I need help. So this is done with the neuron, and then it needs to produce ions in a situation where you shouldn't produce by just waiting, which will be the passive problem, passive transport. You will wait until diffusion sends, or transports a given amount of charges. This is very inefficient transport. Diffusion is slow. This is something we will see in the next class. What is the speed of diffusion? If you want to reach A to B by diffusion, you will go square root of t. If you go ballistic, you go like t, so you go much faster. But to go ballistic, you need something that is giving you a velocity. You need a pump. You need energy. That's the main point. So you rely on fluctuations, you have a very slow, very cheap transport, because it doesn't spend energy, but very slow. And there are always trade-offs between energy and precision and so on. We will discuss all this in the next lecture. Okay. Any questions from the other side? Okay, I think I have to stop. Thank you for your attention.